Okay, let's get started. We're going to talk about kidneys. Yay! Hooray! So the stuff that we're going to talk about kidneys, their filtration and reabsorption of certain solutes. We're going to talk about what's called renal threshold. We're going to talk about clearance. We're going to talk about all the different kinds of blood flow regulation. And then we're going to talk about osmolarity of the concentration, basically what determines the concentration and how yellow your pee is. Really cool, super interesting actually. But before we start all that, we need to know some of the anatomy of the kidney. So I have here drawn a 100% anatomically correct kidney, as you can tell. So this red chunk in there is one of your renal arteries. It goes in, it perfuses your kidney. Interestingly enough, so you have your abdominal aorta coming down here, and about 20% of your blood that's sent through your body ends up going through the kidney with each beat. So your kidney will filter around like 180 liters of blood per day. So a lot of blood.
Excellent.
Super cool. Blue thing is the renal vein. Stuff goes out. Awesome. The pink thing right there, that's your ureter. That's where all of the filtrate and stuff that your body doesn't need anymore, all the waste, gets sent into your bladder and then you pee it out. Awesome. Cool. If you were to follow, say, the ureter all the way up, it gets into this area. This little like cup-shaped thing is called the renal pelvis. It's kind of like where everything just goes into the kidney. Right here is what's called, each of these smaller areas, I guess small but big areas, is called a major calyx, or calyces if there's multiple. And then they branch off into smaller ones, and those are called minor calyces. And then they get to this black line that I have drawn so well, and that's called the renal papilla. And so it gets into here, and this is where all of your nephrons are. And this is my demonstration of what a nephron, quote unquote, looks like. So we have the cortex.
Anatomically correct as well.
What?
Anatomically correct.
Anatomically correct as well, yes. So out here on the edge of the kidney, we have what's called the cortex. And this is where these little bulbs, we'll talk about what those are called later, sit. And then I'm going to do a green dashed line. So like right here. So top is the cortex, and down below it's what's called the medulla of the kidney. And so the medulla is mostly where like the collecting ducts are. That's what this is called, is the collecting ducts. And then that all joins up together into each of the calyces, major calyces, renal pelvis, and then the ureter dumped out. So this area is called the medulla, and then this area is called that was weird. That area is called the cortex. So if we blow up one of these dinky little lines, it's going to look like either one of these or one of these. So you have two kinds of nephrons in your body. 90% of them are these ones. So these are your cortical nephrons. If we look at the line, the majority of it is in the cortex. You only have a little bit in the medulla. For these ones, which are called juxtamedullary, juxta means next to. So this is the outer medulla, and then this is the inner medulla down here. So majority of the nephron is next to the medulla, but some of it is also within the medulla. So we call it the juxtamedullary. So that's these.
Remind me about the nephron again.
The nephron is the major unit of your kidney filtration. We'll talk more about it.
Oh, okay.
So now we're going to label some different parts, except I'm going to use correct colors. So this red line here represents, so your artery that goes in kind of follows the same pattern, breaks off in a whole bunch of little itty bitty branches. When it starts coming into the nephron, we call this the afferent arteriole. And then it gets in here, it's called the renal corpuscle or the glomerulus. Renal corpuscle represents like the whole structure, everything that's inside.
Right.
But that's the glomerulus. And then it leaves still as an artery, and then we call it the efferent artery. sorry, efferent arteriole. Arterial because it's really small, but still carrying oxygenated blood. Well, away from the heart. Not always oxygenated. Great. So, then once it gets past this point, so I'm going to draw a little line. That's this area. And then all of these little squiggly lines are what's called peritubular capillaries. The capillaries is what does all of the exchange with the nephron inside the lumen to the rest of the blood, and then if we like it, we keep it, and it goes out here.
Oh, sorry.
Okay, sorry about that. Okay, continuing onward, as we got interrupted, but that's okay.
How rude.
So we have the peritubular capillaries. So we have all the same stuff on the juxtamedullary one, but down here, because it's so long, we have another area, and this is called the vasorectum. And so this is a part of the capillaries that kind of just like down here, that loop part of the nephron. Now we're going to name all the different parts of the nephron because it's awesome. This area right here, that bowl there, that's called Bowman's capsule or the renal corpuscle. Again, that's where the majority of the filtration, that's where all of the filtration happens. Let me get rid of that line. Okay, there is a section right here that is immediately after the Bowman's capsule. That's called the proximal convoluted tubule. That's where the majority, we'll talk about reabsorption. That is where the majority of the reabsorption of all of our solutes are. So if we push, say, like a glucose into the nephron, we want some way to bring it back into the blood because we use glucose as an energy source. So that's where the majority of things like that will get pushed back into the blood. And then after that, we have a portion that's called the proximal straight tubule. This is where we pull in the majority of our chloride ions. Really useful. Yes?
Is there, for the last one, was there any reason why it was called convoluted?
So you can't tell in my drawing, which is anatomically correct. But in reality, it's like you've got this, and then you've got the part that comes out, and then it's like, and then you've got a little straight portion, and then it goes down. That's a liquid nitrogen machine. It's actually super cool. But that's why it's called the proximal convoluted tube, because it's actually like really weird and wiggly stuff. And the nice thing about that is because it's got so much surface area, you can do a ton of reabsorption. So it's actually like God was onto something.
No ways.
Yeah. So after that, after we finish up at our straight proximal tubule, it goes into this, which is called the descending tubule of the loop of Henle. So this section here is called the loop of Henle. So you have the descending loop, and then you have the ascending loop. Super cool. And then here we have the distal collecting tubule or distal convoluted tubule. They do the same thing. Well, same name for same thing. Different names for same thing. There we go. And then you have this area, which is called the collecting duct. Okay. Whoop-de-doo. Cool. Okay, we're going to draw a very brief pretend great. Awesome. So here's our afferent arteriole. Here's our efferent arteriole. Here's the glomerulus, which is an interesting capillary bed, not like any other capillary beds that you would have ever seen in the body. So there are some pressures that are involved in filtering what we have inside the blood. So there's some pressure from the blood. We've got high pressure here and then a slightly lower pressure here. So we've got some pressure trying to move things into the corpuscle. And that's about 50 millimeters of mercury. We just measure pressure in millimeters of mercury. It's fun. So then we also have some pressure that was already inside the corpuscle that's wanting to get out. And we'll use a different color for that one. That one ends up being about like, oh no, it's not 30, just kidding. This one's about like 10 to 15. And then, if you remember within the blood, you don't because you weren't there, but when you watch my videos, like and subscribe. You have proteins. We're going to use another different color because I have so many colors. This is great. Remember that water wants to flow from area of low solute concentration to high solute concentration. Want, quote unquote. And so you'll have some kind of pressure that pulls water, hence pressure. And this one's going to be around 30 millimeters of mercury. So if you add all this up, you will end up getting a net pressure of about 5 to 10 millimeters of mercury force pulling stuff into the corpuscle.
Right.
So those are the pressures involved in pulling things out of the blood into the nephron.
Okay.
Talk about some more anatomy here. So there's a membrane in between these two. I'm going to use blue, and if I remember correctly, this is called it's either the basement membrane or the basilar membrane, and it's what's called fenestrated, so it's got little holes in it, and that's what allows solutes to flow from the blood through this membrane into your corpuscle. Another interesting thing, I'm going to use the pink for this one. Oh yeah. So what these pink things are what's called podocytes. So because capillaries normally only experience around like 30 millimeters of mercury of pressure, they're not really meant to handle 50 millimeters of mercury. Because if that were true, you'd likely experience a huge amount of edema within your kidney. So what we have is we have these podocytes, which are kind of like structural cells that keep the capillary from bursting and pushing more of that pressure into here rather than pushing out. So super useful. We're going to erase that. An important thing to note is, so that blue line, that membrane that I drew, has holes that are only about 70 nanometers wide, and it is very negatively charged. So what that causes is you can only have things that are small and either have a weak charge or a neutral charge to filter into the kidney. So you've got, say, like large proteins like albumin, that's not going to be able to make it into the kidney, so it kind of just flows through the blood because we want to keep that in the blood. Same thing with red blood cells. They're around like 700 nanometers big, so you should never find blood in your pee. Otherwise, that means you've got some kind of problems here within your Bowman's capsule where red blood cells can just pop through those holes that must have been exacerbated. So healthy kidney, you'll never find blood in your pee. Really important. Okay, so that's that. So that covers our filtration. Basically, I'm going to make a list of a lot of the things that we filtrate. So we've got sodium, potassium, chlorine, calcium. We filter a fun thing called bicarbonate. We filter water. We also filter glucose, lactate, amino acids. And then we filter another thing called just metabolites, which are just like general waste products from cells either dying, going through metabolic respiration, all of that fun stuff. And those things get filtered in, and then they just kind of get pushed through the whole nephron, and then you pee them out because we don't want them. So we're going to talk about reabsorption of a lot of these things. The biggest thing that gets reabsorbed, I guess the most major component of the stuff that gets reabsorbed is sodium. Sodium controls a lot of the reabsorption of a lot of the other things. So this is what the cell on the lumen of your nephron looks like. So your nephron has these circle, and they're just all along the tubes. So as you travel within different areas of the tube, the reason why they have different names is because of the different proteins and the slightly different structure of what they have. So we're going to talk about the proximal convoluted tubule, so just this little early section right here. So the first thing that we're going to talk about is sodium. So right here we have the great classic sodium potassium ATPase pump. That's generating a fun concentration, pushing sodium into the interstitial fluid and then eventually into the blood. Okay, the next one that we're going to talk about is, we have another fun one. Nope. We have a hydrogen, sodium antiporter. We have a sodium glucose, sodium shorthand amino acids as AA, sodium, and lactate, and then I'll do some more. Ah, yes, and water. H2O. Interestingly enough, so there's two different ways that stuff gets reabsorbed within the kidneys. So it can go through what's called a transcellular pathway, which is where it goes into your apical cell and then goes out and is pumped out into the interstitial fluid and then into the blood, or it can go what's called paracellular and go and slip in between the cells. So, of the many, many proteins that we have along our nephron cells, so sodium is driving the majority of this, like all of the movement of these metabolites that we want to keep. So because of this sodium-potassium pump, we're generating a high concentration of sodium out here, and so a low concentration of sodium within the cell. And because you have a low concentration of sodium in the cell, sodium will want to rush into the cell. So here we've got inside of the nephron, this is inside of the nephron. So this is, we're going to label this in green. So we've got the lumen of the nephron, and then out here we've got the interstitial fluid and the blood. Gosh, this marker is getting destroyed. Sorry, marker. Okay, so you have a high concentration of sodium in here because sodium is a small particle, doesn't really have that strong of a charge, so it can pass through that basement membrane and that fenestrated membrane super easily. Basically, a whole lot of it gets filtered. But we really like sodium. Sodium is what helps drive a lot of membrane potential changes. Really useful. Sodium is great. So you have a high concentration of sodium out here and a low concentration of sodium inside the cell because of this pump working. So sodium is going to move in here. And the great thing is we're going to be using secondary active transport. And a lot of them are going to be symport. So what that means is we're using the energy of sodium going down its concentration gradient to either pull things that we want in, or in the case of hydrogen, push it out. And so that's what happens with things like glucose, because we really like glucose. So glucose gets pulled in with sodium, and then glucose will travel through some glucose receptors and then go into the blood. Same thing will happen with lactate and with your amino acids. So all of those big things that we really like, super useful for our body, we want to keep those in the body. We use sodium to pull them in. And so in the proximal convoluted tubule, we will pull in 100% of these. That's where all of that reabsorption happens. Really, really nice.
So it just puts it back in.
Yeah, so blood comes in, it gets pushed out through that membrane, and then because they're really useful to our body, we have a ton of receptors to pull them back into the blood so they can circulate throughout the body.
Okay, yeah, makes sense.
Because of all of the times that we use sodium for all of these transports, we end up pulling about 70% of the sodium that gets pushed into the lumen, we pull it back into the blood. So 70% of the water is water will follow sodium. Water follows areas where there's high concentration of solutes. We're pulling stuff out of here, putting it into the interstitial space and into the blood. So the blood will have, in the moment, a theoretically higher solute concentration. So water will flow from the low to the high. And so we also pull out, interestingly enough, about 70% of our water. And that will pass through paracellularly or go through aquaporins, go through into the blood eventually. The cool thing is that when this happens, water pulls with it potassium and calcium ions. This is what's called solvent drag. As the water moves through it, like literally drags these with it into the blood. So that covers our sodium, potassium, calcium, and water. Okay. Any questions about those?
No.
Great. Okay, so we're going to get rid of those. Now we're going to do a fun thing. We're going to redraw these cells, and we're going to talk about another part within the proximal convoluted tubule. That wasn't too bad. And I actually don't need that. Okay, so now we're going to talk about a fun thing. This is how we take care of bicarbonate. Bicarbonate is a really important buffer in our blood. Our blood needs to stay of a pH of around 7.35 to 7.45. It's a really small zone, and if it goes past that, it's really bad. Not good. So, we have bicarbonate that gets pulled into the nephron. And because it's an important buffer, we need some kind of way to push it back into the blood so we can buffer the blood pH. So we have this fun thing called carbonic anhydrase. I'm just going to abbreviate it as Ca. So what that does is, remember, we have this sodium hydrogen antiporter. Carbonic anhydrase will take the bicarbonate and the hydrogen and turn it into water and CO2. The water follows the rest of the way that all of the water goes. So it's going to go through aquaporins or just between the cells. Great, awesome. CO2, because it's a small nonpolar molecule, it can basically just go wherever it wants. So this is going to eventually flow from here, and it's going to go like inside the cell. Inside the cell, we're going to put it right here. We have another carbonic anhydrase. So it's going to go through here, and it's going to get turned back into that HCO3 minus and H plus. So basically we break it down, turn it into its small components, and then we move it into the cell, and then it gets turned back into bicarbonate and hydrogen. And then hydrogen gets pushed back into the cell and then out. So I don't really change the concentration of hydrogen out here, but we're adding more bicarbonate. So we're being able to buffer our blood more.
Oh, interesting. That's interesting.
Okay, sorry about that. Where's my marker? Red, blue, green. All right, cool, I have them all. Okay, so that's how bicarbonate is buffered.
Very cool.
Great. Cool. And that's most of the things that happen within the proximal convoluted tubule. Really, really important thing. Later on, we're going to talk about how sodium and chloride is moved here within the what's called the ascending loop of Henle. Well, the ascending loop, ascending tubule, part of the loop of Henle, where sodium and chloride are pumped out, and then here water is pumped out. But the majority of sodium, water, potassium chloride, those are done within just this small section of the proximal tubule. Okay, so now we're going to talk about chlorine. Chlorine's super cool. It's exciting. So we're now no longer in the proximal convoluted tubule. If you notice, we didn't talk about chlorine that much. Chlorine is taken care of out here. Interestingly enough, we still have this sodium hydrogen antiporter in the proximal straight tubule. We also have a interesting thing that pulls chlorine in up its gradient as we pull out. Well, as we're pulling in positive charges, chlorine is attracted to be inside the cell. And there's also a high gradient of chlorine from outside into here and then out to there. So chlorine wants to go in. So the interesting thing is, so we have, I'm just going to abbreviate this as HA. So these are some kinds of acids that are produced within the body. And so those are broken down and turned into the hydrogen ion and the rest of the acid. Whether that is, doesn't really matter. So we just call those the anions. So the hydrogen ion is pushed through the sodium hydrogen antiporter and pushed out into the lumen. The large anion is pushed through a chlorine anion antiporter. So the chlorine is moved into the cell and the anion is pushed out. And then the hydrogen and the anion are pushed back together to make some kind of weak acid, which is then now neutrally charged and just removed out, peed out. Great, because we don't need it. And so then we just have that constant cycle of hydrogen moves into the cell from outside of the blood, because we have both sodium and hydrogen antiporters. We also have potassium and hydrogen antiporters. So those pull hydrogen in, the hydrogen here moved out, joined up with these, peed out. Great. So we get rid of hydrogen, we get rid of these anions, and we get to move chlorine back into the body because chlorine is really, really useful. Okay. So that's our proximal straight tubule. Awesome. Okay. Now, the next thing that we will talk about. This is the descending tubule. We're gonna have to draw. I'm gonna get myself some little notes here. So I'm starting at 300, 400, 500, 600. Okay. We're just gonna leave that as blood. ICF blood. Cool. Let's draw our cells again. Great. So this is now the descending tubule or descending loop. So that's this section. So what makes this one different than the last stuff we talked about, it doesn't have any of those protein channels that we talked about. So there's no movement of ions whatsoever, except our sodium, potassium pump. So that's always working in the background. And we have some leak channels, but we don't have any of those major things that's moving this stuff. And so the interesting thing is that this will pull water is going to travel through, because the only stuff that's really here is aquaporins. And so water leaves from inside the lumen, goes into the blood, because the blood has a higher concentration of solids. We moved a ton of stuff out there. And so as water leaves, but none of the solids in here can, effectively what we do is we increase our osmolarity. Osmolarity is the number of solute particles over liters of H2O. So I think you can appreciate that as we decrease our H2O, our osmolarity increases. And so that's what each of these numbers are. As we travel further down the tube, we're pulling out more and more water, but we're not pulling out solutes. So as we keep going down, our osmolarity keeps getting higher and higher. And so at around this point, at the bottom of the loop of Henle, we reach an osmolarity of around 600, which is twice as concentrated as what the blood is normally, because normally our blood osmolarity is around 300. So that's our descending tubule. Now the fun part is now we're going to talk about the ascending tubule. We're just going to abbreviate that as AT. This one is kind of like the exact opposite. So we have our cells, whoop-de-doo, but I'm going to draw a really interesting thing. These cells, unlike the cells in the descending loop, are much tighter packed together, and the water that's still out here cannot, I'm going to draw a little sad face, water can't exit the lumen and go into the blood. So we're keeping our concentration of water the same.
Cannot or is less common?
What do you mean?
Like
water can't leave.
It just can't.
Yeah, no, this area is impermeable to water. So most cells within your body have aquaporins. So like water can like go in and out and reach what's called isotonic state, so where it's like equilibrium of water.
Right.
In this area, we don't have any aquaporins. The junctions between each cells are basically non-existent. So water can't pass through, so impermeable to water. But what we do have is we have a sodium, potassium, and 2 chlorines. This is called the NKCC channel. Moves the sodium, moves the potassium, moves 2 chlorines. Cool. That will pull these in, and then they'll pull out. And so now what we're doing is now we're starting to decrease the amount of solutes here, but not change our water. And those are all going to end up in the blood. Draw little lines here. Boop, boop, boop. Cool. And so now what that does is that starts decreasing our solutes, not increasing this, decreases osmolarity. So we're going to start going back up. So we're going to go from like a 600 to a 500 to a 400 to a 300. And all the way up here, we're going to end up reaching around like an osmolarity of about like 75.
Oh, really?
Yeah, so it gets
way down.
Yeah, so it gets like super, super dilute, even more dilute than where we started. And it's just because of this transporter, we're just pulling in more stuff and water can't go anywhere. Really, really useful. And so one of the more interesting things is, so what sets up this concentration gradient is this channel. So when we pull these out of the cells, they're in that interstitial space. And so we get a huge accumulation of solutes outside of the nephron. And so water wants to leave because it wants to go to that area of high solute concentration because it's way higher outside than it is inside the nephron. So water leaves, and that's what sets us up in becoming these equal amounts. If that makes sense.
Yeah, it does.
Okay. Great. Now we're going to talk about the distal collecting tubule. This one is a lot of fun. And when I say fun, I don't really mean it, but it's exciting. our two. So this time we have sodium can come in. We've got chlorine that, oh, I forgot about talking about maculodensa cells. That's awkward. We're going to talk about those.
That sounds complicated.
No, they're actually super cool.
Cool is not the opposite of complicated.
You're not wrong. Okay, so we're actually going to talk about maculodensa cells. So these are special sensitive cells that are nearby the glomerulus and the afferent arteriole. Yeah, anatomically correct draw.
Of course.
And so they have sodium and chloride channels, and they're able to sense, quote unquote, sense the amount of chlorine that is being filtrated. And so, in essence, these sense how much blood we're pushing through the kidneys, how much we're filtering, by how much sodium and chloride is getting pushed into them.
I see.
And then we'll talk more about the maculodensa cells when we start talking about what regulates blood flow. Okay. Now we're going to talk about the distal collecting tubule. So they still have this area where water can't pass through. And they're also allowing some solids to come through, not a ton, doesn't really change too much. So they'll still let some sodium in, and they'll still let some chloride in, but no water. But the interesting thing is inside they have kind of like this little, at least this is the way that I like to think of it. We're going to draw this. So they have aquaporins on the outside where it's connected to the blood, but they don't have any aquaporins on the inside or the apical side. This is the apical side, this is the basal side. Why it's called that, I don't know why. So down here on this end, there's no aquaporins, but we have this ring where we have a bunch of these aquaporins that are kind of just ready to go into that membrane, but can't yet. And so in the presence of a hormone called ADH binds to this, it activates a G protein, which allows this, the way I think there's a little ring holding structure, allows for aquaporins to go into this area and allows water to come in into here. Otherwise, water will just bounce off, whoop-de-doo, and water will be sad. But if it can go through, water will be happy.
Are you saying water doesn't have emotion?
No, it's literally 3 molecules. They don't have emotions. I barely have emotions. I've got trillions of molecules. And that's an understatement. Trillions on trillions on trillions on times like a bajillion.
I think it's like something to the 26th, anyway.
It's a really big number. Okay, so we've hit 75 milliosmoles up here. So it's super duper dilute. If we don't have ADH, H2O can't come in, and we're pulling in maybe only a couple more solids, so we just continue to have a really, really low concentration, and maybe we'll end up leaving at like 30 milliosmoles at the end. So we have like a super duper dilute concentration, and this is when your pee is clear or really light yellow. Really, really nice. In the event of us being, say we're like super dehydrated, we want to try to keep water in our body. So what will happen is we'll end up releasing ADH. ADH will put these aquaporin channels into the membrane of the lumen cells. Water will come in and we will maintain water within the blood, but what this will do is this will start to increase our osmolarity. And actually we'll end up following the same osmolarity of these things because they're coming in, we have a lot of solutes here, Remember, the solutes, as they leave this area, they kind of just spread out as they go into the blood.
Yes.
So water is going to try to reach equilibrium. So in the presence of ADH, we're going to increase to 300, and then 400, and then 500, and then 600, and so on. So that's when you have like.
And so on. How high does it go?
We'll get to that.
Okay.
So in the presence of ADH, our concentration will go really, really high because we're pulling out more water into the blood because we're dehydrated. And so this is when you have like your super duper yellow pee, really, really concentrated, not great.
Yeah.
Okay. That was a lot to talk about. We kind of actually already talked about these. We'll just cover those really quickly. So this describes what's called diuresis or an anti-diuresis state. So in the event of us not having ADH, we have enough water, we're like, we don't really need any more water. That's called a diuretic state. So we're just shunting water. We stay at that super low concentration of P. Everything's great. As we start to become more dehydrated, we'll switch to start using ADH, and we'll switch to what's called an anti-diuretic state, where we're retaining more water and trying to keep it reabsorbed within our body. So that's what ADH does. We'll talk about ADH when you talk about the RAS system, which is a, what's the word? It's like ATM machine. It's a redundant C. Anyway, for juxtamedullary, because they go deeper into the nephron, they actually reach a even higher concentration. So down here, they're going to hit like 1,200. And say around here, it's going to be like 1,000, and then like 800, and then like 600 to match up with where the cortical nephron is. So because they go lower, they reach a significantly higher concentration. And this, there's a lot of uric acid that messes around with here. Urea is a important metabolite. We both like to keep it and also get rid of it. It's why we call, at least I think why we call urine, urea, and also uresis. Lots of really interesting words, fun connections. So in juxtamedullary, we have all of the same stuff as in the cortical nephron. Goes through, as it gets down super low, we'll hit a 1,200 milliosmolarity. And then we'll go all the way back up, down to around that 75. And then in the presence of ADH, we'll again drop super low, retain a lot of water, super duper concentrated P. This is really, really useful for animals that live in like deserts. So for us, we have 90% cortical and 10% of these. So when we're peeing out, we're only peeing out 600 milliosmol concentrated P. So we need a lot of water just for this to be happy. Things like, well, I shouldn't say things, animals like kangaroos, kangaroo rats, camels, they have way, way more of these than they do of those. And so these are really, really good at retaining water because they get so low in their concentration. So all along here, we've got blood vessels, and in the presence of ADH, it's just pulling out tons and tons of water. So they don't need to drink nearly as often because they're retaining so much water. Super cool, actually.
Very interesting.
That was like one of the coolest things I've ever done. Okay, so that covers the majority of our osmolarity. Talked about kidneys. We're going to talk about clearance, GFR, and renal threshold stuff. You ready for a graph?
Absolutely.
Great. So GFR stands for glomerular filtration rate. Essentially, that's telling you how much stuff you're filtering into the kidney per minute. And the hard thing is depending on what substance you're looking at, that can change. So say if we're looking at something, actually no, that's clearance. GFR, we just want to figure out like what is pumping, how much stuff is the kidney pumping through. So we needed to find something that could get really, really close to that. And that had to be something that was only filtered. It couldn't be reabsorbed. It couldn't be secreted. What secretion means is you pull it from the blood back into the nephron. And it had to be a small chemical, and it couldn't be charged because of that basement membrane that it would have to pass through. And so we found a really useful chemical called inulin. Sounds similar to insulin, but it's not. Inulin. Inulin. And it actually meets all of those things. So then we tried to find some chemical within the body that met also all of those characteristics. And there is one, and it's called creatinine. Similar to creatine, but it's spelled different, so it's different. And so those things go into the blood, into the kidney, they're filtered in, and then they just go out. Super useful. So we're going to draw a fun graph. And so this right here brackets mean concentration. So this is the concentration of, we're going to say I, concentration of, actually we're just going to say X. Plasma concentration of X. When we have something like inulin, as we increase the amount that's in our blood, it increases by the same amount that's in the urine. And so this accurately reflects the glomerular filtration rate. So how much stuff is being filtered in the glomerulus. And so overall, this ends up being approximately 125 milliliters of solution per minute. There are some things where, as we increase our concentration in the plasma, we don't find any in the urine until some point, and then it ends up being parallel to our line for inulin. I'm going to label this one inulin. And then there are other things where, as we introduce them into the blood, very, very high concentration of them in the urine, and then eventually reaches some other point. And so these bends are what's called renal threshold. So all of those different proteins that we were talking about, when it comes to secretion, as we pull things into the lumen, we have a limited number of proteins. And so there are only so many proteins. So if we flood our blood and our system with a ton of these chemicals that interact with them, we can saturate all the proteins. And so where it is in the nephron or in the blood will determine whether or not it's, like whether it ends up in the pee or we just keep it in the blood. So this blue line represents glucose. So glucose is something that we want to keep in our body. If you remember, in the proximal convoluted tubule, we end up keeping 100% of glucose that gets filtered into the kidney. And so all of those protein channels within the nephron, if we add so much glucose that those get overwhelmed, they become saturated, glucose will just pass right on by and stay in the nephron, because that's the only place where we have those transporters for glucose. So it just stays there, and we end up finding it in the blood. And so this point is called that renal threshold. And normally you can't hit that point because we've got so many of those proteins where we can deal with, like, I think it's like 300 grams of glucose per deciliter of blood.
Wow.
Why we use deciliters, I couldn't tell you. I haven't gone to a hospital yet and figured out why the heck we use deciliters. But at this point, right here, once it starts to not be found in the plasma and start to be found in the urine, that means we've overwhelmed those protein channels. And this is a sign of diabetes. Normal person, you will never find glucose in their blood. But if you're diabetic, where say you're type 1 diabetes, you don't break down glucose. So you have a ton of glucose in your blood, way more than you ever should. So then, really, really high concentration, you find glucose in the blood. Makes perfect sense. Something like this, we can call this PAH or penicillin. Really interesting. So when we find a ton of it in the urine and not a lot in the plasma, so that means it's being filtered, it's not being reabsorbed, so it's not getting put back into the blood, but any that made it past the filtration is pulled into the nephron through secretion. Secretion pulling things out of the blood back into the nephron. So some of it passes by, and then even more of it is pulled into the nephron so we can dump it. But same thing with the glucose, there's only so many protein transporters. So if we pump a ton in there, eventually we'll get to a point where it's in line with inulin because we've saturated those proteins. So the cool thing is if we see some kind of number for GFR, or in this case clearance, and when we talk about clearance means how much of it is basically peed out per minute. How much of it is produced in the kidneys, pushed in here, found in the ureters, found in the bladder. So for something like inulin, where it's only filtered, not reabsorbed, not secreted, not excreted, we'll find 125 milliliters per minute. If there's something that is, I should have drawn that in a different color. If we find less in the urine, that means there's more in the blood. So that means something like this is more likely getting reabsorbed than it is getting secreted, and we're keeping it in the blood. And then the opposite is true. If it's above the 125, say 125 of this that's it being filtered, and then the extra 75 is it being pulled in through secretion. So that's renal threshold clearance versus GFR. GFR is a glomerular filtration rate. Clearance is how much we're finding, how much is being cleared from the blood and put into the urine. Oh, great. Okay, I'm going to pause because I want water. Okay. My throat was hurting. It's getting dry from talking so much. Okay, and that's on video. Super cool. Okay, the next thing we're going to talk about is blood flow regulation. This is a long one. There's a lot of stuff that happens. We're going to hit the myogenic reflex first. So, in the event of really high blood pressure, say you're working out a lot and your blood is going really fast, really high heart rate, really high blood pressure. When your afferent arteriole starts to stretch from that high blood pressure, there are stretch receptors in it that will release calcium and cause the muscle to contract to maintain that blood flow that we need within the glomerulus, because we only want around 50 milliliters of mercury. We don't want any more, we really don't want less. So the myogenic reflex, the arteriole starts to get stretched, it will contract to maintain that pressure. ANP and BNP. So ANP stands for atrial natriuretic peptide, and BNP stands for brain natriuretic peptide.
Brain as in brain?
Yes, because we found them in pig brains.
Wow.
But, I mean, there are some neurons that we have that produce it, but the atrial natriuretic peptide is made in your atria of the heart, so the top chambers, and then the BNP is made from the ventricles, bottom chambers of your heart. And so when they get stretched, they will send these signals to the afferent arteriole to dilate and the efferent arteriole to constrict. So we're trying to keep more stuff in the glomerulus to filter out, so the ventricles and atria aren't forced to stretch as much. We have paracrine. So this is paracrine covers what we call our tubuloglomerular feedback. So that goes to our macula densa cells. So when you have high pressure, you're gonna end up getting a high filtration rate. If we have a high filtration rate, that means you're going to get more sodium chloride. When they have a ton of sodium chloride where they're overwhelmed with it, they release which is the potent vasoconstrictor. They will release nitric oxide, so NO. So they're going to release that, and that is going to cause the afferent arteriole to constrict. So when that constricts, that's going to reduce the amount of blood that is in the glomerulus, which will then reduce the filtration rate. And so then the macula densa cells won't have nearly as much sodium chloride, and they'll be like, all right, cool, we lowered it to what we want for homeostasis. On the other hand, if this starts getting really low, you're going to have a low amount of blood in the glomerulus, low amount of filtrate. Macula densa cells are not going to have a lot of sodium chloride. And so when that happens, they're going to release adenosine. And outside of the nephron, normally adenosine will also vasoconstrict, but within the nephron and for the afferent arteriole, it's going to vasodilate. And so when this dilates, we get more blood in the glomerulus, which means we have higher pressure, more filtrate. More filtrate means more sodium chloride here. Macula densa cells are, quote unquote, happy because now they're at a higher GFR. And so that's the paracrines. Sympathetic nervous system, in the event of really low, in the event of really low blood flow, your sympathetic nervous system will start to go into, your sympathetic controls the fight or flight. And so what it will do is it will cause the release of epinephrine. So epinephrine or adrenaline is going to activate your adrenergic alpha-1 receptors, which causes systemic-wide vasoconstriction. The interesting thing is that will constrict both your efferent arteriole and the afferent arteriole, but they will preferentially bind to the efferent arteriole receptors, those alpha-1 receptors. So they bind to there, they start constricting, so you end up getting more blood in here. You'll constrict the afferent arteriole a little bit, but not as much as the efferent. So you'll actually end up maintaining more blood in the glomerulus and increasing filtrate. We have things called baroreceptors. Those will do a similar thing. So baroreceptors are receptors that are found in your carotid sinus and then on your aortic sinus. And so they detect stretch. When they are stretched, they will send signals to your brain to end up causing a decrease in the release of ADH. If you remember, when we have ADH, we're pulling blood out of the nephron, pulling water out of the nephron into the blood and increasing blood volume and therefore increasing blood pressure. So if we decrease the amount of ADH, that ends up leading to more water retained within the urine, less water in the blood, not having blood volume as high, lower blood pressure, and so those receptors aren't as stretched. So we have a lower blood pressure. And therefore, how that affects blood flow regulation here. We have osmoreceptors. This is a really interesting thing. So normally, we have a I'm going to draw it this way. because this is the way that I make sense of it in my head. So normal osmolarity is around 300. And so we have specialized neurons that are really close to blood vessels, and they also sit at around that 300. And so they recognize they can sense the different osmolarities between the two. So if you start having a lot of water pulled out into here, you're going to start increasing the osmolarity of the blood over time. And so if this increases to like 310, this is going to cause an EPSP, or excitatory postsynaptic potential, and it's going to cause this cell to release ADH. When we have ADH is going to pull more water into the blood and therefore decrease the osmolarity.
Makes sense.
And bring the solute concentration back to what it should be. And then if this gets too low, this sends less ADH, this will increase. Cool. So that's our osmoreceptors. And then the big one is the RAS system.
That wasn't the big one?
No,
Oh.
this one's the big one.
Okay.
So, I'm going to draw it out. I'm going to pause the video, and then I will come back when it is drawn. Okay, got it down. There's a lot. So, in response to low blood pressure, your kidneys, there are cells here that are called juxtaglomerular cells.
Hold on.
So, let me use a different color. Here, we're gonna be little green guys. So we're gonna be right there. And they release renin, or renin, depending on how you pronounce it. Renin will enter the blood, and it will find a protein, an inactive protein called angiotensinogen. Angiotensinogen is a protein that is produced by your liver. It's just kind of like pumping it out, among other things.
Right.
When renin interacts with angiotensinogen, it cleaves it and turns it into angiotensin 1. Angiotensin 1 goes around the blood. When it gets to the lungs, your lungs have a high concentration of what's called ACE, or angiotensin cleaving enzyme. So it cleaves angiotensin 1 and turns into angiotensin 2. Ooh. Angiotensin 2 does a buttload of stuff. Really, really useful thing. So the first thing that we're going to talk about is it will flow up to your brain and activate an area called the posterior pituitary gland. Pituitary gland, really useful, don't care right now. The backside of it, really useful, has those neurons that we talked about where they release ADH. It causes them to release more ADH. When you have higher ADH, you pull more water out of this into the blood, keep water. That causes higher H2O reabsorption, super concentrated P.
Right.
It also causes your adrenal glands to release aldosterone. So right above your kidneys, you have a gland called your adrenal glands. This is where we get the word from, like adrenergic or adrenaline, because they make adrenaline. They also make aldosterone. Aldosterone causes your cells. So if you remember, we talked about they have sodium and chloride transporters within these cells here.
Mm-hmm.
In the presence of aldosterone, they will increase their activity. So you'll pull out more sodium and more chloride. They also increase the activity and production of your sodium-potassium pumps. So they pull even more sodium out of the lumen into the blood, and they actually push potassium out of the blood and into the cells. So there's the potential where they can create a hypokalemic environment and that just means a low potassium concentration in the blood. So if you just have aldosterone for a really, really, really long time, not great. But in the short time, really useful for keeping a high concentration of sodium and a concentration of water, because water is going to follow the sodium. If there's ADH. Angiotensin 2 also causes increased activity of the sympathetic nervous system, which activates alpha 1 receptors, which causes constriction of all of your arteries, which will increase blood pressure, but that also increases constriction on these. If that increases constriction on the efferent arteriole preferentially than the afferent, increase this, push more stuff out, higher concentration of P.
Nice.
Interesting. And then it also actually ends up leading to you have an increased salt desire, so you're going to eat more salty stuff, and you're also going to get thirsty, so you're going to drink more water.
Increased salt desire? That's interesting.
Yeah, right?
Whack.
Super cool, because this ends up leading to increased sodium chloride reabsorption, because if you're eating more salt, you're getting more sodium chloride in the blood. If you have more sodium chloride in the blood, you have more sodium chloride here, which is what we want, because we need sodium, we need chloride.
Yeah,
super important
makes
ions.
sense. It's just interesting that happens.
Right?
Huh.
And so the cool thing is ADH stands for antidiuretic hormone. So it keeps you in antidiuresis, you're pulling water out. But it also has the other name of vasopressin, which implies pressing on your vasculature. So it constricts your arteries, which increases your blood pressure, which interestingly enough, when you constrict your blood vessels, increase blood pressure. If you're pushing more water into them, you increase blood volume, which increases blood pressure. Really cool that your kidneys have things that change your urine concentration, but also your blood pressure.
Dang.
Super useful. And so that's ADH and aldosterone, and that controls your osmolarity, all of the blood flow regulation stuff. And yeah, that's it.